Welcome to Ankara University International Webinar Series. I'm Özge Cemiloğlu Ülker from Ankara University Faculty of Pharmacy, Department of Toxicology. And I'm also Vice Coordinator for Internationalization of Ankara University. Today I am here as the moderator of the session. Thank you for joining us. It's a great honor for me to introduce our distinguished speaker, Dr. Helena Hogberg from Johns Hopkins University, Bloomberg School of Public Health. She is Deputy Director of the Center for Alternatives to Animal Testing. First, I would like to thank you, Helena, for accepting our invitation and taking time out of your busy schedule. Johns Hopkins University has played a critical role during the COVID-19 pandemic. And in terms of drug and vaccine research, development research, we know that non-animal methods getting much more importance. And I know Center for Alternatives to Animal Testing has activities from this point as well. I'm so excited and can't wait for listening Dr. Hogberg's presentation. But before that, I would like to give a brief information about Dr. Hogberg. She's received her PhD in toxicology from the physiology department at Stockholm University, Sweden in 2009. The scientific work during her PhD was performed at the European Center for the Validation of Alternative Methods, European Commission, ISPRA, Italy, and aimed to develop new in vitro approaches to detect chemicals with developmental neurotoxicity potential, with a focus on gene expression and electrical activity recordings. Together with Center for Alternatives to Animal Testing Director Thomas Hartung, she started the current laboratory as a postdoctoral fellow in 2010. She was later promoted to a faculty position and was appointed deputy director of the center in the beginning of 2016. Her current research activity is still in the field of developmental neurotoxicity with the use of emerging tools such as 3D organotypic cell models, induced pluripotent stem cells, and transcriptomics and metabolomics approaches. Today, Dr. Helena Hogberg is going to talk about Johns Hopkins University Center for Alternatives to Animal Testing activities during COVID-19 pandemic. Stage is yours, Dr. Hogberg. Thank you.
Thank you very much, Özge, for that introduction. I'm going to start to share my screen. Okay, so yeah, thank you. It's a pleasure to be here. It's a pleasure to see you again. It's a long time no see. I mean, Özge was actually with us for a period of time and did some research with us, and we have just submitted a paper together, so that's exciting. So I'm very excited to be here and tell you about our approach and what we have been doing, because everybody, of course, has been affected by the COVID-19 pandemic, so us, and I'm going to guide you through a little bit of the way we have been handling the situation. But first, I want to give you a background on CAT, the Center for Alternatives to Animal Testing, which is the information and communication hub. So basically our goal, our mission is to really get communication going between different stakeholders. This means academia, industry, and regulators, as well as animal welfare organizations or NGOs. So CAT was established in 1981. And at that time, with a $1 million grant, which was a lot of money at that time, today, not so much, unfortunately, but it was a grant from the Cosmetic, Toiletry and Fragrance Association. And the aim for the center was to encourage the adoption of alternatives to the use of animals in biomedical and pharmaceutical research, in product safety testing and education. And if you are more interested of reading more about CAT and CAT's activity or sign up for our newsletter, I just put the web page there in the lower bottom. So what I said, we are kind of an umbrella organization. We're trying to bring the stakeholders together. So, for example, it's very difficult for industry to talk to regulators on a regular basis, and because it's just not possible for the industry to get that kind of communication. However, if we are going together, industry and regulators under an umbrella and together with academics, we can really start to do dialogue and communication. So we're organizing a lot of workshops with all these stakeholders to try to move the alternatives to animals for use. So our aim is, as I said, promote the alternatives to animal testing where they're not fit for purpose. So I'm not saying that animal testing are never good, because there's a lot of good animal testing done. However, sometimes they're just not fit for purpose. Like in the case of this cartoon here, it's showing a t-shirt and says, these t-shirts were tested on animals. They didn't fit. So when they don't fit, we need to find new approaches. We need to find alternatives. So we are promoting the 3 R's. So what are the 3 R's? Well, these two gentlemen here in the bottom is Russell and Birch, and they wrote a book in 1959, which is called The Principles of Humane Experimental Technique. And that's where they established the 3 R's. The 3 R stands for replacement, which is don't use animals if a non-animal method exists. That can answer the scientific question at hand. Of course, that's not always possible. So then we have another R is reduction. If you must use animals, keep the number to the minimum necessary to answer the question. And this means that you need to really think through your experiments before you conduct them. So you have enough animals to answer the question, but not using more than that. And then finally comes to refinement, because of course, sometimes animal testing is possible, or you have to do it. And then you must use if you're gonna use animals, keep any pain or distress they experience to a minimum. And this is not, the 3 R's is not really about just animal welfare. Of course, that comes with it, it's a good cause, but it's really to do better science. Because if you have a stressed animal when you do your research, that means that you cannot really trust the data that comes out of it. So it's very important to keep this in mind. We're not doing this for animal welfare, we're doing this for doing better science. So CAT has several different programs how we try to achieve this or work towards this. And this is just showing some of these programs, and I'm not going to go over all of these programs. However, I'm going to touch on a few of them and how we have adopted our program during now the COVID-19 pandemic. So I'm going to talk a little bit about our grants and awards program and how we have changed that. Our education, which is of course something that had to change dramatically. And then I'm going to talk a little bit about our own research, which is on microphysiological systems and systems toxicology. So our programs are not only research-based, some of them are, but some of them are also translational. So really how to apply the research we're doing into the bigger field. So how it can be applied and make a change and be used. So here we come to the pandemic. So like many of you, we have also been in quarantine. It has been since mid-March. This is just a picture of one of our Zoom meetings. So like many of you most of us, we were working from home, but we still meet on a regular basis. We have conference calls and so on. And this is one of our meetings, our weekly meetings with the team. I put some smileys here, and this is over the faces of some children, because working from home comes also with some challenges. Not only animals, it can or not only children, it can also be animals. So many of us have young children, including myself. I have 2 small children, and the schools are closed, daycares are closed, so we are all trying to be at home. So if you get interrupted here by my 2-year-old, I apologize in advance. Hopefully they're still sleeping. But of course, this is a little bit of a challenge, and you have to adjust everything and try to make a balance between family life and work, and perhaps change your schedule a little bit, so it's doable. And so, yes, I said we have been in quarantine, but we have still been very productive, of course, with some adoptions. Some of us have even been more productive. So one of our students actually gave birth to her child, her second child, during the pandemic. So that's going to be a very interesting story for him to hear later on in life. Mm-hmm. But we are also starting to open now. So as many other places, so US is a little bit behind in the pandemic. It came a little bit later to us, and we also have not the same control yet of the cases, unfortunately. But we are opening up, and the states are very different. So Baltimore, which is in Maryland state, we are one of the later stage. We're very precautionary here, so we just started to open lately, and it's also different in different areas of the state. So our university is in the city, and we are actually a little bit one of the latest ones to open up. But we will opening up the labs for activities, reduced activities, I will say, actually next week on Monday. So I am in the committee on how we're gonna proceed to go back to the lab, and it's a lot of precautionary. So there's only a few people that can be in at the same time, so we will have to work in shifts. We have 1 person per 400 square feet, which is roughly 40 square meters, if you're more familiar with that. So it's most of our labs are actually smaller than that, so you can actually only be 1 person on site at the same time. We're also trying to adjust how we are going to do with elevators, for example, and stairs and break rooms, because of course, sometimes we're supposed to be there only for doing the research activities, to do the experiments, but some of them will, of course, take longer time, and you need to eat and drink during this time, which is of course also important for your health. And then you need some kind of break room, and these break rooms are often shared. So we're now trying to figure out how we can set up a good schedule so people are not interacting with each other and keep the social distancing. And we are wearing masks all the time. That's also in all the states. You need to wear a mask if you go outside, if you go shopping, for example, and so on. So also at work. And it's I've been in the lab during this whole time, and I'm going to show you the reason why later on. But it is a little bit tiring to walk around with the face mask on all the time. You get a lot of respect for the people in the medical field that are wearing them for 12-hour shift, maybe even longer sometimes. So yeah, it's different, but hopefully, we're gonna adapt to this new norm. So I just wanted to show this map. So of course everybody knows this map because it is from Johns Hopkins University. It's not from my lab per se, or from our group per se, but of course we are working very close together. So, and I know this is the map that most people have used, and most people are using at the moment. This was taken a screenshot for a few days ago, and you can see it's a lot of cases and a lot of death. Especially in the United States, you can see there's a lot of red here, which is scary and challenging. And I think one of the reasons we have a lot of cases here is also how people are interacting with each other. So me coming from Europe, being Swedish, so I have really the two difference here, right? Because you're probably very well aware. I know it has been reported a lot in the news that Sweden is very open, you know,
Yeah.
and we also have a lot of death there. But saying even if it's open and the, you know, we can still, daycares are open, schools are open, and so on, we have been encouraged to not have social distance, to have social distance in Sweden, and people are actually following those recommendations. On the contrast, in US, we have, you know, regulations in place. We're not supposed to gather together, but Americans in general, they want to decide on their own. Not talking for everybody, of course, but that's very often, you know, this is how the country is built up. Yeah. So sometimes it's hard for people to really follow the guidelines or the recommendations from the authorities, because they want to decide on their own. But I hope, I mean, it's in some states it's worse than others, yeah, and hopefully we will get under control with it also here in the United States. We also have, of course, a lot of poverty, and that's some areas that, you know, have been more affected than others, unfortunately. So this map is maybe not as famous as the other one, but I just wanted to put it out that there's a lot of different maps on the same webpage, and I just put on the link here. So this is supposed to be United States, and these are the different states, and you can see that this is, are we flattening the curve? So if it's more green, that means that the cases really are going down. If it's red, the cases are still going up. I'm myself is in Maryland, which is here. So you can see our cases are going down. So it seems to be doing fine here. But there are some states like Michigan, for example, where the cases are still going up. Yeah. So that is, it's concerning. And we will see now when we're opening what will happen. Yeah, that's, but okay, this is in United States. Maybe it's not so applicable for you unless you're going to come here and visit, which I don't even know if it's possible at this time, but when it opens up again. But we also on this site, they have also for other countries. I didn't see Turkey here, but I think if you scroll down, you can probably look at all of the different countries. But what you can see here is, of course, US. Everybody knows we have
most affected.
Yeah, but and now it's Brazil coming, right? So that's what there's a lot of unfortunately death and cases in Brazil. Also there, you know, in the poor areas. So how is Turkey doing, actually, just for?
Yeah, Turkey is. Yeah, we have now. We opened all the. We are getting normalization. So, and the cases are. Still, we have around 1,000 cases, but it's like a flat, so not increase or not decrease these days, just flat.
Okay, well, that's good. But and you keep the social distance and so on, right?
Yeah, social distances, masks, and hygiene is 3 very important points.
Yeah.
For protect ourselves and our loved ones. So
yeah, no, exactly. I mean, that's the thing. I'm you're not so maybe so scared about yourself that you will get really sick, though there are young people also that get sick, right? But we all have someone close by or friends of a friend or so on that are older, that are, or people that are in risk. We need to, those are the ones we're trying to protect, right? So yeah, okay. So so then, what have we done at CAT with our program? So I'm gonna give just a couple of examples because I cannot go through all of them, but this is our grants and awards program. And the reason I decided to show this is because it has changed some. We have due to COVID, but I also thought this could be of interest for you because you are also eligible to apply for our grants. So everybody looks for funding, right? So in this case, we are, it's a small grants program, so it's not a lot of money, but it's provide funding for science to develop alternatives to the use of animals. And it's 40,000 for 1 year. And even if it's a small number, we have been giving out about 300 grants and that's in more than $600 million. And what our awardees are saying often afterwards is like, okay, it's not like a huge amount of money to do a big project, but it can take you from start to rethinking, like if you're doing only animal experiments and then you see, maybe if I can actually answer these questions with a non-animal method in the same and get the same kind of results, that people can start to really think that way and start to develop something in their own labs. So as I said, this is open, it's global. So you are also very welcome to apply for this. Unfortunately, the deadline for this year has already passed, but there's something to look into maybe going forward. And we would be very excited to have your applications and hopefully you can get funded.
Yeah.
But so one of the things, and that you mentioned as well, so COVID research in COVID is a lot of people are using animals, and for us it was I felt a little bit, everybody was at home and you see people dying around you or get very sick and you want to contribute somehow. You want to, what can I do? I felt, very sitting at home and useless in one point, but who cares about my little research when people all around the world are dying or getting really sick? I wanted to help somehow. So what we decided to do was to actually dedicate some of our grants program also to COVID research. And I was, it made me frustrated because I got these emails like, almost every day from different providers saying, oh, now animal research is even more important. We need to use more animals. This could be a good mouse model for looking at COVID. And I felt frustrated because, animals actually, they are not affected the same way as humans by COVID. It came from an animal and they didn't have at all the same kind of problems as humans have. So this is really a human disease. And what you're normally doing when you want to make an animal model for viruses, this actually goes for most viruses, like HIV, for example, Zika, animals don't get infected the same way as humans get, or actually get infected. So what you're doing is that you immune suppress your animals. And that's how you get, the immune system is so suppressed, and that's how they can get infected with the virus. But we know that the immune system, especially when it comes to COVID, plays a huge role. And so then to use an animal model without that kind of immune system makes me wonder if this is really a good model for this. I'm sure for some research it can be useful, but I don't think we will understand the mechanism and maybe also not come up with a cure if we don't use a more human-like model, so that they're really mimicking what is happening in a human situation. So we decided, as I said, to dedicate some of our grants program to a fast track program for research on non-animal approaches to understand the mechanism or come up with medicines and vaccines for coronaviruses. We said coronaviruses instead for COVID because, COVID is the problem we have right now, but who knows what will happen in the future. I mean, this is probably not going to be our first time, unfortunately, that this is happening. We know, people have talked about it for a long time. The pandemic will happen and it will probably happen again. So more models, more that can really predict human disease will hopefully help us in the future. So this was also a good opportunity for us to reach out to our sponsors. And we got some of our sponsors and some new sponsors actually to put in money into this program. And of course, funding, a lot of companies are not doing great. So, and our organization is actually sponsored by industry, for example, and some of them have not been able to pay this year because of problems in their own organization. And I think that's also something that a lot of businesses, not only also universities actually will have a financial struggle at the moment. But this was something that the Humane Society International and Animal Free Research, they felt that this was something that they really wanted to help with. So they helped us to co-sponsor this grant opportunity. Again, the deadline has unfortunately passed, so you cannot apply, but there will be maybe more of this coming out. So, but we were really, normally for our grants program, we have about 30 applications. And in this case, which was a short, it was only like, a month possibilities for people to apply. And we received more than 60 submissions. And we are currently reviewing them. And unfortunately, we don't have enough money to fund many of them. So we're looking into fund maybe one or two only with our budget. So of course, it's, the chances to get funded is very small, but hopefully we can also encourage more of our sponsors to put in money into this program specifically, and then we could potentially fund more. So then I'm going to talk a little bit about the education, because of course education has also changed a lot due to COVID. These are just 6 courses that CAT is involved in, which are 3 of them are in toxicology. One, the animal in research, is a refinement course, and then we have 2 courses in epigenomics. So 3 of these was actually online courses from the beginning. So they didn't change much, right? Because they were already online. But the other 3, or the other 2 were actually in class. So those courses have now been done online instead. So I know a lot of people are doing this, and of course it happened really fast, from one week to the other, we suddenly had to do it online. So I think we learned a lot on the go, and I think it has worked nicely. I think we still need to come up with new tools, especially I think the discussion and the interactions with students. It's a little bit challenging with online. I feel this, now I have you there, it's really great, but you don't see the people you're talking to in the same way, and the interaction. So I think there we need to start to develop some new tools. I know on Zoom they have, for example, breakout groups and so on, but I think as we go, we will learn more, and we already have some training in hand now, people that are more familiar with this and come up with new tools, because this is gonna unfortunately continue. So we already talked before here that you also have all your courses are for the fall also going to be online, and the same for us. We don't see that we're going to go back to the school until after Christmas. So the rest of the year is going to be online, and you'll see by then. I mean, some people are saying also until we have a vaccine, we will not be able to do this. There might be smaller online classes, like 10 people or so, but for most of our classes, they will be online. I'm wondering about my daughter too. She's going to start, preschool this fall, and I don't know what will happen,
Yeah.
with that, and so one is starting preschool and the other one is starting kindergarten. So yeah, we will see. Hopefully there will be, because it's also challenging, right, for people having their children at home. So my oldest daughter, she had some Zoom classes with her teacher, but it's challenging because she's only 5, so she kind of needs our support, and I have to work, and then we have a 2-year-old, and then it's hard to help her doing school activities and then take care of another little one, and then at the same time work. So there needs to be a lot of flexibility in life at the moment.
Yeah,
I can just imagine people that have older kids, maybe 10 or so, that needs a lot of support. My 5-year-old doesn't. I mean, it's mainly talking kind of. So but yeah, but I think there's a lot of tools up there, and it's not impossible. But some of our courses, 2 of our courses here, also freely available on Coursera. And I wanted to put this out there in case some of you would be interested of also participating in these courses. As I said, they are for free. Coursera is actually really good. There's a lot of nice courses there. We only have 2 at the moment, and these are in toxicology. So this is Tox 21 and evidence-based toxicology. So if you search for toxicology at Coursera, these are these are the 2 first hits that comes up. Actually, there's not a lot on Coursera toxicology, but this I also think is going to change now with all the online teaching, that maybe there will be more courses available. And so if you're interested of learning also from other universities, I recommend you to go look here. So our 2 courses here are, as I said, the first one, Tox 21C, is to introduce students to the paradigm shift in regulatory toxicology. So it's really talking about moving away from the traditional animal experiments to alternative approaches. It's explaining some of the shortcomings with the current systems that we have, which is the animal models, and then how you can apply new novel technologies. And here I'm talking about 3-dimensional cell cultures, for example, computational work like AI, also high throughput, and it's a lot of different teachers that are lecturing. So it's both from regulators and industry and academics, and how you can apply these new tools in toxicology. And then the other courses, this one we have by now have about 4,000 students enrolled, and actually because of COVID, it went up quite a lot during this time. So it was a lot of students, more students than normal, have signed up during COVID. So again, something good. And as I said, they are free unless you want to have a certificate from it. Then there is a certain fee. I think it's $50 or something. I don't really know, but it's, you can also just take the courses if it's only for learning. Just do it for free. That's and then the evidence-based toxicology. This is it's talking about the evidence-based medicine, which maybe people are a bit more familiar with, and then how to translate it into toxicology. It's also teaching how to do quality scoring of studies and doing systematic reviews and meta-analysis. And this course, even if it's, says toxicology, I think this is a really good course for a lot of people to take, especially to be, critically reviewing different studies, and also to learn how to report yourself on your research. Because so it's, even if it's based on toxicology, I think everybody would benefit from learning these things. And systematic reviews is something I'm really something I think is really amazing now, because it's not, a normal review is when you just do your bias interpretation of all the data that you think fits to the purpose. A systematic review is really when you have to take into consideration all literature, and then you have to do some kind of a scoring. So some studies, of course, higher or more likely to be true than others. So it's a new way of, and it's time consuming, but I think that's really going to move the field forward. It will help us to combine a lot of information, but also to learn where the gaps are and where we need to put into more research or more resources. And this course, I've had about 2,000 students this far. This course started, I think, 1.5 or maybe even 2 years after the other one. So that's probably one of the reasons we have less students here. But I mean, if you're interested, you can just, click through. But there, as I said, Coursera also have a lot of other courses. So I think, I really recommend people to go there if you have the time. So a success story then, because COVID, it's a lot of tragic things about COVID, but there's also some really good things. And so we organize workshops and conferences. So this is one of our workshops or conference that took place last week, which is the 3R symposium. And we couldn't do it in person, of course, so we had to do it online, which was actually a success, because normally we have maybe 50 participants, and now we had 180. So, and from all over the world, yeah. So because people would not necessarily come to this meeting in person, but when it's online, more people, you know, have the opportunity to really take them. So that was really, I think, something really good coming out of this. And we did a survey afterwards, and okay, only 100 of the 180 participated in the survey, but 83% of them actually said that they want to participate online next time. So we have decided for next year, if we can have it in how, or, you know, on site, we will also do a hybrid, so people will also be able to join this online. And I think we also had our board meeting actually, and my boss has always been against having board meetings remotely because we have talked about it before. But I have to say it was really a success. More people were participating than before. The discussions were pretty good. So I think it gives us new opportunities to like, you know, explore new ways of doing things. And I think that's never bad, you know, just to think outside of the box and start to change.
Yeah, I think so. Yeah, I'm also thinking about these are an opportunity for us, not traveling, but we can, you know, just listen and learn many things.
Yeah. No, and it's also
Because
a lot of things are recorded, so you can actually look at them afterwards. So
yeah.
this conference was also recorded, and the presentations will be available in the fall. That's why I put up this the web page. If you're interested of any of the work we are doing, or want to see some of our lectures and so on, you can sign up for our newsletter. I have it, I think, on the last slide, if people are interested, but you can also see it through our
Thank you, Helena. Thank you.
So coming up now, we have a summer school in innovative science without animals that's taking place now in June. It was also supposed to be at Hopkins, but now we can't. So it's going to be online and it's free. So if you're interested in this, so I said COVID made it, we have to do it online. So this is something also, if you're available or interested, you can sign up. What this, it's actually together with the physicians committee. So they are the main organizer and we are just hosting it. And it's also in collaboration with the Joint Research Center, European Commission. There is a summer school done at the Joint Research Center in Europe every year. I assume it will not take place this year, or if maybe it will be some kind of an online version as well. And we felt that this would be something, or the physicians committee and us felt that this is something that should be done also in US, because unfortunately US are sometimes a little bit behind, especially when it comes to animal use and refinement. So what this course is, or this summer school is supposed to be, is for students and early career scientists, but actually any person could benefit from this one. And it will give you information on in vitro, in silico, and humane-based methods and approaches for both toxicology and medical research. There will be 2 different tracks, one in toxicology and one in biomedical research. So there will be different, there will be just lectures, virtual laboratory tours. There are also going to be e-poster presentation and some kind of engagement and communication between the speakers and attendees. So I know we have mentors, for example, so going to be small work groups where you can sit and chat and just meet the scientist in person. And if you participate, you are eligible for a poster and participation award, and you will receive a certificate for your involvement. But if you don't have time to follow the whole summer school, it's quite some days. I'm teaching 3 lectures, by the way, so
So the program, yeah.
But I love this to teach students, because they are so, you know, interested and they ask a lot of questions, and that's really what makes you excited. And you come back, you know, they're so happy and so enthusiastic about things that it really gets back to you, and you feel, oh, this is why I'm doing what I'm doing. So it's very nice. But if you cannot follow the whole, you can also just take some lectures if you want to through Zoom. So you can sign up on this web page, or you can also go to our web page and you will find it through there. So if you're interested, you can do that. So the final things I wanted to touch on is our research work. And we're doing research on the brain, mainly in our group. That's what we're mainly interested in. And why are we doing the research? Because current in vivo approach does not necessarily predict human health effects. So this is a little cartoon showing from the drug discovery that you have some screening methods first in the company where a drug passes. And then if it passes, it goes into the animal experiment. And when it passed the animal experiment, it goes into clinical trials, so in humans. And there, unfortunately, most of the times it fails. So 89% of the drugs actually fail in clinical trials in humans. And this is due to either they had effect in the human that were not seen in the animal, or it's a lack of effect in humans that you did see in animals. So it's, you know, either it's not safe in humans, or the efficacy of the drug is not there. So as I said, our group is focusing on the brain. That's my main interest, and there the failure rate is even higher. So we have 94% failure rate for neurological therapies. Some diseases, such as like Alzheimer's disease, it's higher, 99.6% failure rate. So there is no drug that cure you from Alzheimer's disease. There are about 5 drugs that are on the market, and none of them, you know, some of them are having some kind of good effects, but most of them also have a lot of other effects. So like
side effects, yeah.
There's no good drug for Alzheimer's disease, neither to cure, absolutely not to cure, but also not for coping with the symptoms. So of course, this means that it's very costly and very time consuming to bring a new drug to the market. So what we want is to perhaps speed up and be sure that something is more safe before it goes in the clinical trials, and also save money. So we say sometimes fail early and save money, because if you don't go to the animal experiments or do all these tests, which is very both time consuming and costly, I mean, at least, you know, you don't spend so much money, and then you can perhaps have some other kind of approach. Eventually you probably need to do some animal experiments, but it can be, you can, for example, figure out what species to use, a species that is maybe more similar to the human, and doesn't necessarily have to be a monkey. Actually, it's just some models are more predictive of humans than others when it comes to certain effects, right? So sometimes a rabbit is a good model, sometimes it's a mouse, sometimes it's a rat, sometimes it's a monkey, sometimes it's none of them. So, and we just need to figure that out before. So our approach is to use human organotypic cultures. So we're using 3-dimensional neural systems. And the reason we're using 3-dimensional neural systems is because, well, all organs are challenging to model in vitro. So with cell cultures, but that's especially for the central nervous system, because the central nervous system, the brain, is the most complex organ in the human body. And it consists of numerous different cell types. So it has different kinds of neurons, for example, as well as glial cells. And these cells have a very complex neural network and architecture. So it's very difficult to mimic this architecture in a monolayer, simple in vitro cell culture. You kind of need a 3D structure, because in a 3D system, you have cells that higher cells of interaction, and they are biochemical signaling, and that's crucial for the function of the brain. So without the connections, that's the purpose of a brain, to have good connections and to interact with each other. And some of these processes, such as myelination and synapse formation, for example, they are essential for the function, but that you also need to have a 3D structure to be able to mimic that. So that's why 3D models are getting more and more popular to use. And I think especially for the brain, it's important. So our group have developed a 3-dimensional model of human brain development using induced pluripotent stem cells. This was a collaboration with 3 organizations. So it's us, CAT, at School of Public Health, and then it was School of Medicine that's very good in stem cells, as well as Kennedy Krieger Institute that knows a lot about neurodevelopmental disorders. So the photos here is the 3 people that have been mainly involved in the work, the initial work with this. So it's the PI, Thomas Hartung, myself as the co-PI of this grant, and then David Pamies that has been doing a lot of the development of the model. So the goal was to develop a human mini brain, as you can see here. What we're using induced pluripotent stem cells, and it's just showing a little cartoon how we made the model. So we differentiate induced pluripotent stem cells into neuroprecursor cells, NPCs, and we do this in monolayer culture. And then we scrape the cells off for single cell suspension, and we are culturing them on this shaker that you can see here, gyratory shaking. And due to this shaking, the cells are clustering together and form a 3-dimensional structure. So it's a very simple model, actually, in that sense. It was simple to make, then it's pretty complex, as you can see, it's 3-dimensional. So what does this brain model consist of? It has all different cell types in the brain, more or less, all neurons and glial cells. It does not have microglia, which is the immune cells of the brain, but you can add them, and we have successfully added microglia to this model. The size is pretty small, as you can see, 350 micrometer, and it's important to not have too big of a size, because if it's too big, then oxygen and nutrition will not go to the core of this spheroid, and that's make necrotic and apoptotic cells, so dying cells. But what we have shown with our model is that it's very reproducible in size and in cell composition, so it's have more or less always the same amount of neurons and astrocytes and so on. And the beauty with using induced pluripotent stem cells is that you can use different genetic backgrounds. So you can also use cells from patients, for example, with a certain disease, and that can help you tremendously in finding mechanisms on diseases and also to understand vulnerability in certain populations, for example.
Mm-hmm.
And not only are the cells present, they're also functioning. So we have neurons, I mentioned before, the role of neurons are to communicate with each other, which we have demonstrated that they can here with electrical activity recording here to the left. But they also have functioning glial cells, so they have functioning oligodendrocytes, which is one cell type in the brain, and what they are doing is they form myelination, which as you can see here on the picture in the lower right. So how this is myelin sheet wrapping around an axon, you can see here. So it's just showing that, you know, a 3-dimensional culture has more likely to be more functional than a monolayer culture because it looks more like the in vivo situation. So some of the ongoing research and application which we have been using this model for is, you can see here, what we are doing toxicology. That's what we are, you know, mainly our expertise. So developmental neurotoxicity, neurotoxicity, but we're also using this model for other things. We felt like, you know, it's important to try to use this. We have a tool here and see where are the applications. So we have teamed up with experts in other fields. So we know we've used it for neurodegeneration, like Parkinson's and ALS, for example, for multiple sclerosis, which is, you know, the myelination I showed is important. We have done some cancer research, some resilience and blood-brain barrier work. So we're also looking at, gene environmental interactions. So if you are more susceptible to exposure to certain toxicants, for example, we can look at that by using different genetic background. And here, a lot of our work goes into autism as well as Down syndrome. We're doing some inflammation work, and here I come into the COVID again, because we also looked at infectious diseases with our models. We have we have successfully infected our brain model with different viruses such as Zika, HIV, but also now COVID, actually. So even if, due to COVID, we have had limited lab activities, we have been fortunate in a way that we have been doing some COVID research. And because we do that, we have been exempt. So we have still been able to go into the lab and do research. And of course, if you do research on COVID, you can also do a little bit on the side on your other projects. But saying that, we have really had limited activities. We haven't done a lot of experiments that we would normally have been doing. So of course, that's one of the problems. But we are currently infecting our mini brain. And actually, we did submit a paper yesterday. So I cannot go into the detail on the data yet, but it seems that the brain cells can be infected with COVID, which is, I don't know, maybe good for us, not bad, and bad for the rest. But hopefully, it doesn't. There's a lot of more to study here. This is just a first small publication, and hopefully it will be accepted shortly, and you might hear something in the news about this. That's my thinking of this. So with that, I wanted to thank you. This is the end of my presentation, and I just put up, how you can still get access to our both research and all other activities, not only activities by us actually, but in the field. We all, in our newsletter, if there's something big going on in the field of alternatives, or our sponsors, for example, have something, webinars or so, this will be in our newsletters. And so if you're interested, you can sign up for this. I think it's maybe a couple per month that comes out, and you can.
It will work. Yeah. Thank you, Helena.
Yeah, well, thank you. It was nice to
Yeah.
talk about our work.
And thank you for your very informative and also inspiring presentation. I don't know if we have any question. Let me check them.
Yeah, should I stop sharing my screen?
Yeah. How to improve international cooperation in online environment for education and research between universities?
Yeah, I mean, I think I didn't mention this, but we actually already are collaborating with several universities. So CAT, Center for Alternatives to Animal Testing, has the US branch, but we actually also have a European branch at Constance University. So we are in Europe, and we are in US. And as I said, the Coursera I really like, because I think that's something, everybody can have access to a lot of courses. Also, our online courses are available. Anyone can sign up for those from outside of our university if you're interested. Some of the other courses as well. I think they're probably a bit expensive, to be honest. Like, that's why I think Coursera, and for us, CAT, it's different than from the university. Of course, universities, especially in US, they're private in that sense, that they need to bring in money. So I think that's one of the way we are getting money is through our courses and people paying for it, but anyone can take those courses. But for CAT, the mission is really to reach out to as many students as possible. And that's why we are exploring the Coursera. It's nice if people want to get the certificate, but other than that, you know, it's for free, right? So we just want everybody to know. And I think with this pandemic, maybe there will be more collaborations.
Collaborations, yeah.
Because we see how easy it is. Yeah, you don't need to be in the same city to work together.
Yeah, that's true. Yeah.
So,
And one more, what do you think about open science and open data? How can we share research data?
so that I'm very much in favor of open source, and there's a lot of journals now that are open source. I'm the chief editor for the neurotoxicology section of Frontiers, and Frontiers have an open source journal, so it's available to everybody. I think that's really crucial. We cannot limit, you know, that research should not be available for everybody. I think it's really crucial to make science go forward. Data needs to be available for everybody, and that includes negative data. And there's where some area where we really need to improve. You know, I don't know how many times you have felt, it happened to me many times, I feel like, oh, I have this great idea. Why hasn't anyone done this? Well, actually they did, but it didn't work, right? Because those kind of publications never comes out. I also think with the systematic reviews and evidence-based toxicology, you know, there's a lot of bias in us, you know, what we're publishing. It's partly due to us scientists, because we don't want to show weak data, say it like that. You know, if you do 5 experiments and one experiment doesn't fit with your scope, you maybe don't want to publish it, and you decide to take it off because it makes your study seems weaker. Sometimes as a scientist, you actually want to show it, but then the journal comes back and tell you, oh no, you know, I think it's better if you remove this, or the reviewer says, you know, or do 10 more experiments. And of course, we're all on a limited time, and, you know, PhD students are finishing, master students are finishing. So I think we have to be less biased when we are publishing our data, and we need to really be honest with what we show, because otherwise, again, like a lot of research can actually not be reproduced, because, you know, it's not efficient information in the publications. You maybe have left out some of the things, and I think the pressure of publishing is so high, unfortunately, that sometimes a lot of crap comes out, and I think we need to change that.
Yeah, yeah, I agree with the negative data. We need to share the negative data as well.
Yeah, there should be some journal. I know there are some platforms, some internet sites and so on, where you can put negative data, but I don't know how well they are looked at. So I mean, I don't go to them specifically myself, but it would be nice to have, you know, journals encouraging also negative data to publish.
Thank you. Yeah, and I also have a question about the you know, the model, your 3D brain, mini brain model, and you said you can you show that the brain infected by coronavirus. So can it be a risk for, especially for developmental neurotoxicity of the
That's one of the questions we try to answer.
yeah, this,
No,
because it is really serious, huh?
Yeah.
So,
No,
because,
that's one of the things we, you know, where we want to go with this research as well. We want to try to understand that. We know it. I know there's not so many studies yet, right? Because this is new. A lot of these children has not been born yet. The ones that has been born, you know, you don't see this kind of effect early on. You actually see this kind of effects when the children are 2, 3, 4 years old.
years later.
Yeah, well, I mean, some effects maybe you can see ahead of time, but like autism, for example, if that would be of concern, we're not sure about. But what we do know is that COVID does affect the nervous system, because there are studies from Wuhan, for example, where they say that more than 30% of the patients actually have some neurological issues. Why? If it's, you know, primary due to the infection or if it's secondary due to lack of oxygen because the lungs are not functioning. But I feel that we see more and more research coming out now that also other organs are affected. It's not only the lungs. And, you know, the long-term problems with these patients, I think we really don't know yet. Yeah, we don't know also if the virus, it doesn't seem to be clear yet if the virus lays latent in some tissue or in some cells. Yeah, so I think there's a lot of things that will come out in the closest.
Yeah.
I wanted to say also, we also know there's one study where a mother gave birth to a child that actually was also infected by COVID and had some neurological problems. So it's might be something that, you know, related to something else, but it could also be an effect of the COVID. So I think there's a lot of research to look into here and try to understand what's going on. And
maybe we need progressive researches about this issue by following the babies until maybe 3 years old and understand what happened in this
no,
process during the
I think so too. And I think those kind of studies are also hard to do with animals, because animals does not necessarily get, you know, speech delay because they don't have autism. They don't get. There are some animal models, you know, modeling ADHD and so on, but we still don't know if the physiology is really the same in those animals as it is for humans. And so. And the truth is, of course, the humans, right? But it will take long before we have those data. So if we can do some kind of research ahead of time, maybe we can, you know, be
human.
Yeah.
Progressive human research, epidemiologic,
And try to, you know, also come up with how we can, you know, help this if there is a concern. Maybe it's not, we will see. And then you know, but help them. But also I think elderly people that have had COVID, for example, I mean, people are saying, you know, some people are sick for several months, and are really still not coming back. And this can be even young people. So, and what will be, you know, the long-term effect of this? We cannot say today, and maybe nothing. Hopefully not, but you know, I think so. Wash your hands, social distancing, and be careful. I absolutely
Hear my mask. Thank you. Do we have any? No. So thank you, Helena, again, taking your time and sharing these valuable information with us.
Thank you, Eski. It was really nice to see you. It's nice to do this. I'm happy.
Okay. Goodbye. Have a nice day. Take care.
Thank you.
Okay.